Hello, today I'm going to tell you what I used and why for my animal cell model project. For the nucleus, I used half a toy soccer ball because it creates DNA and soccer is in my DNA. For the mitochondria, I used a battery because the mitochondria is the powerhouse of the cell and creates all the ATP, and batteries create energy as well. For the vacuole, I used a container filled with water because vacuoles hold mostly water and waste products. For the Golgi, I use a stamp because the Golgi packs and ships macronutrients to other parts of the cell, so it's basically the UPS of the cell, and that's what stamps go through. For the ribosomes, I use beans because ribosomes are packets of protein, and beans are very high in protein. For the rough endoplasmic reticulum, I use straws and beans because it is tube-like and it is where ribosomes are collected and transported throughout the cell. For the smooth endoplasmic reticulum, I use straws and Skittles because it is also tube-like, and the smooth ER makes lipids, which are fats, and you could say Skittles are kind of fatty. For the cell membrane, I used gray paint because it is a protective barrier and slightly a different color, so I just painted it gray. And then for the cytoplasm, I used the green styrofoam ball because I needed somewhere to put all my organelles, and cytoplasm holds all the organelles in the cell. For the lysosomes, I used ramen noodles because lysosomes eat bacteria, and so do worms, and I thought that the ramen noodles looked like worms. For the nucleolus, I used a marble and a bean because the marble looks like there's something in it, and so it looks cool. And I used the bean to represent a ribosome because the nucleolus makes ribosomes. And finally, for the nuclear envelope, which is the outside of the nucleus, I just used the outside of the soccer ball because I didn't know what else to use, and also it's just a different color than the inside, so I figured it was fine. The 3 different organelle disorders I found are the Krebs disease, Parkinson's disease, and achondrogenesis. The Krebs disease is from a malfunction in the lysosomes. It is caused when a person inherits 2 copies of an altered gene, 1 copy from each parent. The symptoms include progressive loss of vision, difficulty walking, decline in thinking skills, and others. Most treatment is supportive, but a hematopoietic stem cell transplant has been known to work in some cases. Parkinson's is from a malfunction in the nucleus. It is caused when the nucleus in cells that makes dopamine start to die, so then the cells start to die. The symptoms are tremors, stiff muscles, difficulty moving, and many, many more. To treat it, there is a medication that would help, and also so would physical exercise. And for the last one, achondrogenesis is from a malfunction in the Golgi body. It is caused from a mutation in the TRP11 gene, which provides instructions to make GMAP210 protein. That protein plays a big role in the function of the Golgi body. Symptoms include extremely short limbs, a narrow chest, short ribs that fracture easily, and lack of normal bone formation. Most infants die before or after birth due to this, so there is no cure. Bye.